Namaste friends, welcome back to Biology Center for True Learning. In this video, we discuss about the concept probability. As we know the fact that Mendel was not a biologist, he was a mathematician. So as a mathematician, he was applied some mathematical measures or mathematical applications for the explanation of his monohybrid and dihybrid process. So let us see how he applied the mathematical expressions. So what is probability? So in general, probability is nothing but the extent to which something will occur or happen, or the mathematical expression or mathematical measure of likelihood is called probability. So for your understanding, I tell you an example. This is the 1 rupee coin. When we flip 1 rupee coin, so we have 50% of chances to get head, then 50% of chances to get tail side. So this is the probability. So what is the probability of getting the head side when we flip the coin one time? That is 50%. So we can write it as 1 by 2. Then what is the probability of getting the tail side when we flip the coin only one time? So again 50%, we can write it as 1 by 2. So 1 by 2 is the probability of head and 1 by 2 is the probability of tail. So I hope you understood the concept of probability. So let us enter into the topic. So how Mendel applied the probability concept for the monohybrid and dihybrid cross results. Here, so I take one example, that is round seed production. Here round seed production is an event. Mendel was demonstrated the event round seed production in both F1 and F2 generations. In F1 generation, what is the probability of getting round seed production plants in the F1 generation? Here probability is guaranteed to occur. So because all the plants produced in the F1 generation are round seed producing plants in the dihybrid cross. So here the probability is sure. So all the plants are producing the round seed plants. Next, F2 generation. Come to F2 generation. So what is the probability of getting round seed producing plants in the F2 generation? Right. The probability of getting this event, the probability of occurrence of this event in the F2 generation, 3 out of 4. It means, so give a look at this Pinhead square. So this is the Pinhead square of F2 generation in dihybrid cross. Totally 16 plants we obtained in the 4 different varieties. So let me first write phenotype ratio of dihybrid cross 9:3:3:1, right? So out of every 4 plants, 3 plants are round seeded producing plants. This is the meaning of it. Just check it, cross check with this table, right? So round yellow, how many plants? Round 9. Then here 3, 9 plus 3 becomes 12. So out of 16, 12 plants are round seed producing plants. So what is the meaning of it? Right, for example, 3 by 4, 3 by 4, 3 by 4, 3 by 4 is equals to 12 by 16. The results are same. So we obtain the same results in case of using the Pinette square and in case of applying the probability rules. So why and when we go for application of probability? So in case of single gene or two genes involve the cross, the Pinette square is easy and better. So when large number of genes, for example 5 or 6 genes involve the cross, so drawing of Punnett square is very difficult. Some hundreds of squares need to be drawn. So it is time taking and so energy waste of time. So in that case, the probability rules are best and best. Right? So results are same. So this explains the same. So out of 16, 12, round 9, here round 3, 9 plus 2, 12 plants are round seed producing plants. Here 3 by 4 means 12 by 16. Then here the probability may be empirical or theoretical. So this is general point. This probability may be empirical or theoretical. So what is empirical probability? Here the calculation of probability by real life observations. Here the theoretical probability is based on, so it is based on certain rules and assumptions. Here something is going to be predicted. Then based on those predictions, the rules and assumptions are formed. Based on them, so probability is designed. But here the Mendel went for the empirical probability. So based on the real life observations, based on the original observations, the Mendel given the results. Here two important rules can use for demonstration of the results in mathematical form. How many rules are there? Two rules. One is product rule. The another one is sum rule. One is a product rule, another one is a sum rule. Our main focus is on product rule in this video. Right. Actually, these rules can be applied for where events are independent. I mean, these rules can be used for explaining the independent transmission of the characters. Of course, so one thing is very clear. So in monohybrid and dihybrid cross experiments, the independent transmission is there. So we can easily apply these rules. So what product rule says? So what is product rule? This rule says that the probability of two independent events occurring together can be calculated by multiplying the individual probabilities of each event occurring alone. So what the product rule says, the probability of two independent events occurring together can be calculated just by multiplying the two events probabilities, two individual events probabilities occurring separately. Here, so in case of for applying the product rule, the two events should be happening, should be occur simultaneously, should be happen. They should be happen. So in case of independent so events only the product rule applies. Otherwise, so it cannot. So this is the product rule. So let us try to understand this rule with an example. So all of us are very known the dice. So dice has 6 sides, right? So this is the dice, right? This dice has 6 sides. So each side has numbers like from 1 to 6, 1, 2, 3, 4, 5, then 6. Here, so I ask one question. What is the probability of getting the number 6 when we roll the dice once? So once again repeat the question. So what is the probability of getting the number 6 when we roll a dice once? This is the question. So it is very simple to say the probability of getting number 6 is obviously 1 by 6, right? So 1 by 6, right? Then next question, what is the probability of getting the number 6 if we roll 2 dice at the same time? So in right hand we have 1 dice, in left hand we have another dice. When we roll 2 dice at a time, so what is the probability of getting the number 6? So it is very easy to say. So we can, these are the two events, right? So rolling the dice with the right hand refers one event, then rolling the another die with left hand is referred to as the another event. So here these are two independent events, they are not dependent, they are independent events. So by multiplying the probabilities of these two events, so we can get the probability of getting the number of chances to have the number 6, right? So here we are going to multiplying the two events probabilities, right? 1 by 6. So when we roll the dice with our right hand, the probability become 1 by 6. So when we roll the dice with the left hand, again we have the probability of 1 by 6. Sorry. So we have to multiply this. Then ultimately the probability of getting the number 6 when so we roll the two dice simultaneously is 1 by 36. So this is the probability. So it is a simple example. Now we go for understand the same rule by taking the example which is very close to the Mendel experiments. Mendel experiment, right? So another example, that is cross between heterozygous individuals in monohybrid class. Right? So what are heterozygous individuals? Capital T, small t, and capital T, small t. Here the question is, what is the probability of getting homozygous recessive individuals? The question is, what is the probability of getting small t individual in the cross, in the next generation or in the cross? Here, so we need to think that from this parent, this is the, for example, so this is the female parent, this is the male parent. From this parent, the probability that the 50% of gametes can have small t. From this parent also, the half of the gametes can have small t. There is a 50-50% chances of getting small t allele into the gametes from both the parents. So let us calculate the probability of getting homozygous recessive individual from this cross. Right. Answer: probability of getting small t individual. So it is one event, right? So this event occurs so when two events are multiplied. Here two events are, one is the formation of 50% gametes with small t from female. The another event is formation of gametes with small t allele from the male. They are the two independent events. So when multiply, so the probabilities of these two independent events, so we will get the probability of getting the small t individual. So is equals to probability of mother to contribute small t gametes into probability of father to contribute small t gametes. Here I already tell you the 50% of chances are there. So we can write it as 1 by 2. So 50% of chances for capital T allele, the 50% of chances to small t allele. Then we can write it as 1 by 2. 1 by 2 again into, here also 50% chance is there. Then it will become 1 by 4. The 1 by 4 is the probability of getting small t individual. So let us cross check it with the Punnett square of monohybrid cross. So let me draw here. So when we make a cross between two parents on these female gametes capital T small t, so on this male gametes capital T small t are taken. So write down the combinations: capital T, small t, then capital T small t, again small t, small t. Just by looking at the Punnett square, we can easily identify the probability of getting small t. Right? There are 4. Out of four, only one individual having small t allele. That is the pure recessive plant. The answer is 1 by 4. So here also we got 1 by 4 probability. Both are same. Right. Next, so we see the same rule in case of dihybrid also. Right. The same example, cross between heterozygous individuals. The offsprings produced in the F1 generation of dihybrid cross are capital R, small r, capital Y, small y, right? So when we cross between these two, here the question is, what is the probability of getting capital R, small r genotype? Then another question, what is the probability of getting capital Y, small y genotype? Actually, the capital R, small r genotype is responsible for round shaped seeds. Then capital Y, small y genotype is responsible for the yellow color. We know that. So let us find out the probability. Here the formula is probability of is equals to RR into capital Y, small y. I mean the probability of RR genotype and probability of so YY genotype. So separately, so we calculate the probability of RR and YY. Then it is easy to calculate the total probability of so this individual. First, probability of RR. Right. So draw the Pinner square. When we cross two gametes RR, then RR, then RR, capital R, small r, then small r. So what is the probability? So out of 4, 2 are capital R, small r. Then how can we write? Half, right? So we can write it as 1 by 2. The probability of R genotype is 1 by 2. Next, so what is the probability of capital Y, small y? Then draw the Punnett square and arrange the gametes vertically and horizontally in this way. Then write capital R, sorry. So YY, right? YY. Capital Y, small y, capital Y, small. Capital Y, small y, and capital Y, small y, small y, small y. Again, in this case also, two genotypes are appears here. Then the probability of getting YY becomes again 1 by 2. 1 by 2, right? So substitute these values here. 1 by 2 into 1 by 2 is equals to 1 by 4. So 1 by 4 is the probability of 1 by 4 is the probability of getting RR YY in the F2 generation. It means so out of each 4 plants, one plant will be this one, right? One plant will be this one. So you can check it with the Punnett square. So this is the one line has 4 plants. This is also has 4. This is also has 4. This is also has 4. Totally 16, right? Totally there are 16 offsprings. So out of every 4 offsprings, so one offspring is capital R, capital Y, small y genotype, right? So in this line, this one, right? Capital R, capital Y, small y, right? In this line, this one, capital R, capital Y, small y. So out of these four, capital R, capital Y, small y. So out of these four offsprings, so one is capital R, y. So 1, 2, 3, 4. Then out of every 4, the one will be the capital R, capital Y, small y organisms. So here once again I tell you, so you can apply Punnett square method for the cross involves either 1 or 2 genes. It is easy to try, it takes less time. So if large number of genes or 5 or 6 genes involved in the cross, so you have to go for probability. So just I show you one example. Cross is like this: capital A small a, capital B small b, capital C small c, capital D small d, capital E small e. Then this is the genotype of one individual. Here again capital A small a, capital B small b, capital C small c, capital D small d, capital E small e. So if you want to draw the Punnett square for this cross, so you have to draw so many number of squares. It is time consuming process. For these kind of examples, so you better to go for applying the product rule concept. It is very simple. The probability of the whole event is equals to the multiplication of individual probabilities of two events occurring alone. This event occurs alone. This event occurs alone. These two events are not dependent. They are independent. So when we multiply these, we can get the probability of that. So here I tell you one simple thing. So what is the phenotype ratio of monohybrid cross? That is 3:1, right? 3:1 is the phenotype ratio in monohybrid cross. 3:1. So by using this, by multiplying two phenotype ratios of monohybrid cross, so we can get the phenotype ratio of dihybrid cross. That is 3 into 3, 9. Then 3 into 1, then 3. Then 1 into 3, 3. Then 1 into 1 becomes 1. So what is the ratio we get here? 9:3:3:1. Thank you for watching this video.